Okay, hi everyone, and welcome to today's online lab on antimicrobials. This is a rather easy, straightforward lab because a lot of it is just a review of what you've already learned in lecture. So when we're talking about antimicrobials, we're looking at chemical agents to control the growth of microbes. And we've already discussed various different forms. One big concept that I always want you to remember is the difference between disinfectants versus antiseptics. So again, remember, disinfectants are chemicals that you use on inanimate objects, okay? Things like surfaces, tables, anything that's not living. Whereas antiseptics are chemicals used to kill microbes on human tissues, on living skin, okay? So make sure you don't mix these up. We have various examples that we went over in lecture. We had phenols, alcohols, surfactants. If you remember, gaseous agents being the most strong for sterilization. Now, when we talk about those examples, there are a few things I want you to remember. One big thing is about alcohol, because so many times people think of alcohol as, this fantastic antimicrobial, when in fact it has a very big weakness. And what did we say that weakness was? The problem is it evaporates too quickly. Okay, so it's a very short duration on the surface or on the skin, means that it's going to do a less effective job than some other things might. The other term I want you to be familiar with is surfactants. Remember, surfactants means soaps and detergents, but there is a very big difference between the two. With soaps, you're just physically degerming, okay? So degerming, again, means to physically remove, kind of wash away microbes. But what aren't you doing? You're not killing them unless it specifies that it will kill the microbes. Detergents, on the other hand, these destroy the cell membrane of the microbes. So detergents actually do kill. They are very good antimicrobials. Now, no matter which of the antimicrobials you're using, there are a lot of different things, different factors that can affect how good of a job these antimicrobials are going to do. Basically, when we talk about the efficiency of them, we're talking about how good of a job do they do getting rid of microbes. So one thing that influences them is concentration. And in lecture, we talked about the concentration of the microbes affecting the antimicrobials. But concentration of the chemical itself, of the antimicrobial, can also have a big influence on how good of a job it does. So for instance, high concentration. So for example, using 90% bleach versus 10% bleach. How does that influence the efficiency? Well, higher concentration of the antimicrobial means better efficiency, more efficient, better job of killing. Okay, there will be more microbes killed. But you then have to be careful, of course, of the possible side effects to the humans as well when you're dealing with concentration. Length of exposure, we just mentioned a couple of minutes ago. Length of exposure means how long is this antimicrobial up against those microbes. So for instance, if you're trying to clean a table and you just spray Lysol and instantly wipe it up, well, that's not as efficient as spraying the Lysol, letting it sit on there for 10 to 15 minutes, and then wiping it up. Because the longer the exposure, the better the efficiency. Okay, so longer exposure means more efficient antimicrobials. That's why also we said alcohol is not that great as an antimicrobial, because alcohol has a very short exposure. The type of microbe also matters. And when I ask which would be more resistant, there's a few different answers. One of the big answers for this would be any pathogen capable of endospores. they will be more resistant. Also, any of the pathogens that have capsules or, glycocalyx, they're more resistant. Remember, these structures help protect those bacteria. So things like endospores or capsules, that's more resistant. Less efficiency for the antimicrobial. Environment also matters. And when we say environment, we don't mean a bunch of trees outside in nature. We mean the environment around that microbe. So for instance, when we say environment, we can mean things like organic matter, vomit, pus, blood, urine, all of these lovely things that you encounter in the hospitals especially. When that organic matter is present, it will be harder for you to kill those microbes. Your antimicrobials will be less efficient because those microbes are now protected by all of that lovely grossness. In addition to that, those organic matter materials, sometimes they also break down certain antimicrobials. So again, organic matter such as pus, blood, vomit, urine, any of those lovely things, they make antimicrobials less efficient. An environment that would instead make it easier to kill microbes, for instance, might be heat, extra heat, having flame present, okay? That helps kill microbes. Or simply saying, having less organic matter. That's an environment that helps make it easier to kill the microbes. Now, in addition to the antimicrobials we have talked about so far, you also have chemotherapeutic agents. The way these differ from other antimicrobials is that chemotherapeutic agents, where do you use them? Internally. Okay, so remember that chemotherapeutic agents are for killing microbes internally. So you take these as drugs or antibiotics, pills, injections, whatever. You are using chemotherapeutic agents to kill infectious diseases within your body. The two categories of these agents are antibiotics and synthetic drugs. And you may ask yourself, well, how do these differ? Well, you are all experts in antibiotics by now. We've talked about it so much. You already know that antibiotics, where do they come from? Bacteria and fungi, from actual microbes. So even though we may mass produce them, they technically originate from microbes. Whereas synthetic drugs are drugs or medications that are completely synthesized in a laboratory setting. Now, when we do talk about those antibiotics, there are a few things we keep mentioning. One is the fact that bacteria make them, and why would a bacteria want to make something that can kill bacteria? Kill off that competition. Get rid of them, okay? So, in fact, it'll usually kill the ones that are most closely related, most similar, because they share the most common resources. Now, when we talk about antibiotics, you have to be very comfortable with the terms broad spectrum versus narrow spectrum. So the example that I like to give you is regarding gram-positive and gram-negative cells. Because a broad spectrum antibiotic, as you see from the picture on this slide, will kill a whole bunch of different types of bacteria. So for instance, a broad spectrum would kill both gram-positives and gram-negatives. Whereas a narrow spectrum antibiotic would only kill one type, one or two types, okay, very limited group. So for instance, the narrow spectrum might only kill gram-positives, okay? So make sure you're comfortable with those two terms because remember, that affects the concept of antibiotic resistance. You don't want to use too many broad spectrum antibiotics too often. Then we get to the other big term that I keep emphasizing throughout this course, which is selective toxicity. You have to remember what this means, and you have to remember what the examples are. So when we talk about selective toxicity, remember You want to pick a drug that will kill the pathogen without hurting the host, okay? So you want it to be selective towards the exact pathogen, not the host. And because of that, the examples that you should know is what is the most selectively toxic bacterial target? The cell wall, because they have it and we don't. So you're not going to hurt our cells. Whereas the least selectively toxic target would be the plasma membrane, because yes, they have it, but so do we, and ours is almost identical to theirs. So if you target their plasma membrane, you're going to hurt the host as well. Now, when we talk about antimicrobials and antibiotics in the laboratory setting, or even in the medical industry, you're going to want to know, well, how good is that antibiotic? And how good is it at killing each particular bacteria or pathogen? Because as you know, one antibiotic isn't just going to kill every single possible bacteria. There's a lot of variety in terms of different bacteria having different susceptibility and resistances. So in order to evaluate how good a job an antibiotic would do at killing a particular bacteria, we do the Kirby Bauer test, which you see in the figure below as letter A. So that's the test where you swab the bacteria all over the plate, and then you place different filter pieces of paper onto the plate, and each filter is soaked containing a particular antibiotic on it. And you look to see for each one, does it kill the bacteria and have a nice big zone, or does it not kill the bacteria and you don't see any zone at all? When you do this test, you should be using a Mueller-Hinton media. So that means instead of the TSA plates that you've seen us use for a lot of our other experiments, you would use Mueller-Hinton media. The reason you do this is basically it has the same nutrients in it and food sources for the bacteria that you would find in the TSA plate, but it does not contain anything that might inhibit or break down antibiotics. Whereas if you use regular agar plates, sometimes their components will break down certain antibiotics, especially tetracycline. So they would basically, the plate itself would be inactivating the antibiotic. You would see no killing of the bacteria, and you might assume that that bacteria is resistant, when in fact it's just that the plate messed up your experiment. So that's why Mueller-Hinton is so important. It will not inactivate any of the antibiotics. Now when you perform this test, what you then would do is measure the diameter of each of the zones of inhibition. And depending on whether you see a big or small diameter, you would then determine whether or not the bacteria is resistant, intermediate, or susceptible to that antibiotic. And so what you should do is above the word resistant, write down no zone of inhibition. Because remember, if that bacteria resists getting killed, you will not see a zone. If that bacterium is susceptible to that antibiotic, you will see a very big zone of inhibition, such as the one right here, because that bacteria is susceptible to getting killed. It's able to be killed by that particular antibiotic. Now, in addition to the Kirby-Bauer test, which is the one with all these circle zones of inhibition, you also have what's called MIC test. That's minimum inhibitory concentration test. And instead of using a single circle with a set amount of antibiotic, what you do is you use these strips, and on this E strip, each line has a different concentration of that particular antibiotic. So you'll notice up here, there's a much bigger amount of bacteria killed, whereas down here, you don't see any bacteria killed. because this is very low concentration of that antibiotic, and it increases until this is the highest possible concentration of that antibiotic. So this is a very valuable technique because not only does it let you know if that bacterium is resistant or susceptible to that antibiotic, but it lets you know the exact range of concentration that will kill that bacterium. So for instance, right over here would be the minimum concentration that you would be able to use to kill that bacterium, because you notice below that you don't see any death occurring. Okay, this one down here has a much higher minimum concentration, meaning that you need to use quite a bit of this antibiotic before you actually get any killing of that bacteria. So looking at this picture, you would prefer to choose this antibiotic rather than that one, and you would choose a range within the zone of inhibition. Okay, so that's the MIC test, gives you the exact range minimum to be able to use to kill the bacteria with that antibiotic. Now this is just to show you how in the lab setting, once you have your plate with the different zones of inhibition, you would use one of these little rulers and you would count the millimeters. So make note that the millimeters are the little small lines on this side here. So that 1 is actually a centimeter. So that would be 10 millimeters, 20 millimeters, and so forth. So what you would do is you would take the diameter. So you could draw a line from here all the way across to here, and you would measure that line as the diameter. For each one, you'd measure the diameter of the zone of inhibition. And then what you would do is you would look on the chart here and you would check. So for instance, here we have penicillin. So you would go to penicillin here and notice this is Staph aureus. So you would go penicillin when testing Staphylococcus, and once you have your measurement, which here would have been a very small measurement, let's say 10 millimeters, even less than that, you would look over and you would say, oh, our diameter is less than 28 millimeters. So Staph is resistant to penicillin, Staph aureus. Okay. You then look at the next one, vancomycin. Okay, you would go vancomycin, you would measure this diameter. Let's say it's 18 millimeters. You would go vancomycin. When testing Staphylococcus, go across. 18 is greater than or equal to 15, so Staph aureus is susceptible to vancomycin in this case. Okay, so we would use the chart. Now, you're also usually able to, if you are not doing something ultra, important, like you're not working in a medical lab or something, you can also just take a look at this and analyze it in the way of saying, well, looking at it without even measuring anything, this right here is the biggest zone of inhibition. So I could say that Staph aureus, this sample of staph, is most susceptible to tetracycline, least susceptible or most resistant to penicillin, because I don't even need to measure it to visually see the big circles versus the little ones. So if, let's say, I was prescribing a patient from this result, you would want tetracycline or even gentamicin, vancomycin, streptomycin. Any of these would be better than prescribing penicillin to this patient. Okay? This is just to show you exactly how those types of experiments would be set up. So for instance, if you were in lab doing this right now, you would swab your labeled agar plate with a whole bunch of bacteria. So you would pick one bacterial strain, swab it all over the plate, and then you would sterilize your forceps and place the antibiotic discs separated from each other so that they're not mixing together, and so that you can see a nice zone of inhibition. Always being careful not to place them too close to the edges or anything, because you don't want to have trouble looking for those zones later on. You also, in addition to testing antibiotics, would test for disinfectants. So what you do when testing for disinfectants is, instead of using a pre-made disc that has the antibiotic on it, you use blank filter discs. So these little circles that are blank, you take them and you dip them gently into a disinfectant of your choice, such as Lysol, bleach, alcohol, hand sanitizer. Sometimes we use hydrogen peroxide, mouthwash, all different variety of disinfectants and antiseptics. And what you would then do is place those discs on the swabbed bacteria and look to see, do you get a zone or do you not? And for those, you're not even measuring it. There's no, pre-made table. You just simply look, is there a circle zone of inhibition or is there no zone, to see how effective that disinfectant would be. So now we get to all of the results sets. So for these, the exact questions and analyses you will find in the post lab. So we have results set 1. We have results set 2. Results set 3, which is now looking at a patient and only has antibiotics, whereas the previous slides had disinfectants and antiseptics as well. And then you have the final result set 4, which is different strains of bacteria. Notice we have 2 copies of Staph epidermidis, there's Staph aureus and E. coli, all tested with the same antibiotics. Now keep in mind these are real results from students, so notice that sometimes the placement of the discs will not be perfect, so pardon the location on some of them. So for instance, I just want to point out that is tetracycline result right over there. It's a little tough to see, but you see that zone of inhibition over there. Okay, contact me if you have any questions while reviewing these slides, and have a great day.